The abdomen of adult insect represents a large hind tagma of the body bearing no segmental paired appendages except the cerci, styli and external genitalia, mostly confined to the terminal segments. Segmentation 12 segments. This is the third or hind part of the insect body. The segmentation of the abdomen takes place as soon as the segmentation of head and thorax has been completed. During the embryonic stage, the abdominal segmentation proceeds always cephalocaudally. The abdomen of embryo of various insects is segmented into the 12 segments, particularly in the orthopteroid insects, but only proturans possess the well-defined, evident 12 segmented abdomen. In a large number of insects, the abdomen consists of 11 segments, most common during the adult stage. In the higher adult insects, the abdomen is apparently bearing only 10 or even 9 segments. The columbola seems to be exceptional as the abdomen is composed of only 6 segments, and in some cuckoo flies, the abdomen is made up of 3 or 4 segments. The total number of segments of abdomen is maintained throughout the larval and adult life. In counting of total number of segments, not only external segmental plates but some other criteria must be borne in mind, such as the embryonic segmental ganglia, coelomic cavities, segmental appendages, intersegmental sutures, distribution of hairs and bristles, and metameric nerves, muscles, and spiracles. The 10th segment. It is formed in the embryo of these insects of all orders except Protura, Columbola, and Homoptera. It is often retained in the adult Hemimetabola, while in Homometabola it may occur in fused condition with the last abdominal segment. The 11th segment. It is represented generally by a pair of appendages, the cerci in adults of Protura to all Hemimetabola. In Coleoptera, Hymenoptera, Siphonoptera, Trichoptera, and Lepidoptera, the 11th segment is well evident in the embryo, but it is indistinct in the adult. The 12th segment, or telson. Heymans reported through a series of embryological studies that the 12th segment is represented by the dorsal supraanal lobe, epiproct, and the ventral paired subanal lobes, paraproct, as seen in Thysanura, Blatteuria, Odonata, Epimeroptera, Phasmida, Orthoptera, and Dermaptera in embryonic and post-embryonic stages, while they persist in adults of only Blatteuria, Plasmida, Orthoptera, and Dermaptera. The segment is represented by the paraproct alone in Thysanura and Epimeroptera, while Odonata loses these structures completely at the adult stage. In skeletal composition, the abdomen, irrespective of the variable shape, is typically composed of a series of distinct alike segments extending to maximum 12 in number. Each segment bears a dorsal plate or tergum, the ventral plate of sternum, and the lateral pleural membranes. The tergum as well as sternum are devoid of sutures. The pleural membranes also do not possess the sclerites except in Thysanura. In some insects, however, as a result of secondary sclerotization, the tergum bears the submarginal ridge. The antecosta and the anterior most band, the aerotergite. The anterior margins of the terga also produce the epididymal process in some insects. All the structures serve for the attachment of the muscle. The adjacent terga are generally communicated by the intersegmental conjunctive membrane which facilitates the posteriorly directed overlapping. Then pregenital, genital, and postgenital segments. In the pterygote insects, the abdominal segments are commonly referred to as the pregenital or visceral segments to those bearing no appendages, and the genital segments to those possessing the genital aperture and the external genitalia. This is a single genital segment, the 9th in male, while 2, 8th and 9th in the female. Reduction of pregenital segments. The first sternum or its fusion with the metasternum occurs in all orders of Pterygota. In Coleoptera, the first 2 segments reduce to membranes. In Hymenoptera, the first and half of the second segments join with the metanota. The second segment in adults become the petiole as a narrow and elongated constriction lying in between the thorax and abdomen. The fusion of the pregenital segments occur much predominantly in Coleoptera and Heteroptera. In some insects, mostly Cerambicidae and some other insects, the pregenital segments undergo telescopic arrangements that initiates the formation of a retractile tube. It is used as a fine piercing device for laying the eggs in the plant or animal tissue. And abdominal appendages. Each abdominal segment or insect embryo bears a pair of rudimentary appendages, but most of them disappear during post-embryonic development. The adult insects, particularly pterygotes, have the appendages mostly on some posterior segments of the abdomen and a pair of cerci on the 11th segment and genitally and the segments 8 and 9 in female and only in 9 in male insects. These segments are therefore called the genital segments bearing genital organs. The segments prior to 8 are considered as pregenital segments, which are in adult pterygotes devoid of any appendages. In Apterygota as well as immature or metamorphic forms of Pterygota, however, various segmental appendages occur on the pregenital segments of the abdomen and are commonly referred to as the pregenital appendages. The 11th segment of some insects bears a pair of appendages, the cerci, which may be considered as the postgenital appendage. Pregenital appendages. The ventral tube of the adult Columbola is formed due to a fusion of paired appendages of embryonic segment 1 of abdomen. Similarly, adult Protura and Diplura also bear a pair of appendages on the first abdominal segment. They though occur as paired glandular appendages and commonly termed as pleuropodia. On the abdominal segment 1 of embryo, they disappear completely in the Thysanura pterygota with post-embryonic development. Similarly, segments 2 to 7 were paired appendages as legs in the larvae of Neuropteran Cialis and Caesura, and in addition serve as secondary function of the tracheal gills. Similar type of structures are also found in the larvae of some Mecoptera, Panorappa, and some other Hymenoptera and Lepidoptera, in which they develop into paired locomotory appendages. Okay, and Blepharoceraptera, they develop into larval legs known as pseudopods. In Apterygota, however, they are continued to few segments and are widely modified. Then styliform appendages. Apterygota commonly have the styliform pregenital appendages. Thysanura have styli on 2 to 9 abdominal segments, particularly in Machilinidae. The abdominal appendages are in a reduced condition, and each is defined into a limb base of coxite and a distal stylus, but in some cases coxite fuses with the sterna and appendages are represented only by the styli. The styli do not develop on the first segment. Similarly, the 10th appendage has no appendages. Lepisma, however, bears the pregenital abdominal appendages on segments 7 to 8 only. The pregenital segments are provided with a median or paired reversible vesicles. Diplura also bear pregenital abdominal appendages on segments 1 to 7. They are styliform in some insects, but first pair is modified into lobulated appendages in Compoididae. Protura bear paired appendages on the first 3 abdominal segments within a protrusible sac. Then columbolan appendages. Columbola do not bear external genitalia. They however bear 3 pregenital abdominal appendages. The colophore, tenaculum and furcula. They are situated medioventrally and are unpaired appendicular structures in the adult columbola. Then the colophore, the first abdominal segment bears ventrally the median tube or colophore. It is a long cylindrical tube terminating in a bilobed protrusible vesicle filled with blood. The colophore is a movable structure. It is articulated by a pair of retractor muscles. It may function as an adhesive organ. The tenaculum. The third abdominal segment bears ventrally a minute median organ. It is also called the retinaculum or hamula. It consists of a basal corpus and a distal paired process called the rami. The rami contain teeth on its outer margins and internally an adductor muscle. It helps in springing process by holding the flexed furcula. And the furcula, the fourth abdominal segment, bears ventrally a springing or leaping organ as the furcula. It consists of basal median, manubrium and a pair of arms. Each arm is divided into proximal long segment and dense, and the distal short segment, the mucro. The manubrium is innervated by two pairs of muscles, one each of flexor and extensor, adductor and abductor muscles. And larval leg-like or gill-like appendages, the abdominal segments 1 to 8 of the larva of Cialis bear paired lateral long appendages. Okay. Then each leg is six-segmented structure and it is tapering and attached basally to the lateral pleural lobe or coxopodite, which is innervated by the muscles of movement of the appendages. Similar type of segmental abdominal appendages are also found in aquatic Dicticidae and some other larvae of Coleoptera. The paired gills developed on the abdominal segments of ephemerid larvae also exhibit homologous morphological structures through differing in function. Then prolegs of caterpillars. The caterpillars of Lepidoptera bear 5 pairs of prolegs, 4 pairs on the abdominal segments 3 to 4 and the 5th pair in the X segment. The number and development of prolegs is reduced in some species. They are considered as the two segmental appendages and found to be developed from embryonic limb rudiments and perform locomotory functions. Each leg is differentiated into 3 regions: a proximal limb, middle cylindrical tube, and a distal cuticular or retractile lobe, a pad called the planta. It bears the curved claws or crotches. The planta is provided with retractile muscles. The prolegs of X segment are called the anal legs or postpedis and resemble with other prolegs in structure through differing musculature. The larvae of the sawflies, Hymenoptera and some Coleoptera also bear the prolegs. Genital appendages. The genital segments 8th, 10th in male and 9th, 8th in female. They are a group of appendages commonly called the genital organs, and they constitute a well-defined male and female external genitalia respectively. They are involved in the mechanism of copulation and perform sperm transfer in the male while oviposition in the female. The main genital organ in the male is therefore the intermittent organ or terminalia or penis, while in female is the ovipositor. The other genital organs in true sense are the accessory structures according to Snodgrass in 1935. The accessory genital organs can be differentiated into two groups. The organs of one group constitute the median intromittent apparatus and called the phallic organs, and those of the other group, which are the peripheral accessory structures, are called as the periphallic organs. The phallic organs are involved in coitus, while the periphallic organs perform the function of holding or grasping the female during copulation. The accessory genital organs of the male include the paired periphallic organs such as claspers, grasping hooks of the 9th segment, and even the cerci of the 11th segment in some species. The secondary copulatory organ situated on the second abdominal segment of Odonata represent a group of phallic organs and a penis. In Diplura, the penis is rudimentary. In females, however, besides the primary genital organ of the egg-laying tube, the ovipositor, which develops from the appendages of 8th and 9th segments, no remarkable accessory external genital organs are found. The male genitalia, they exhibit wide variation in different orders. The penis, it is formed due to a fusion of the paired appendages of the 9th segment. It is single median tubular structure or a phallus in most of the insects, while paired in Ephemeroptera and Dermaptera. In some higher insects, few accessory phallic structures after establishing a close association with penis constitute a complex median intromittent organ. The median phallic organs are median genital outgrowths of the 9th segment and found around the gonopore and develop either into the lobes and phallomeres or into a median tubular penis or phallus. The phallic organ is formed from 3 phallomeres in Dictyoptera. The typical phallus is a conical or tubular structure but variably modified in various groups or insects. There is only a single tubular phallus in Thysanura and Placoptera. Okay. Phallus of penis is divided into a proximal part, the phallobase and terminal part, the edegus, while in undifferentiated condition, either phallus is called the edegus. The phallobase, the walls of phallus from ectophallus, endophallus and cover outer and inner chambers around the gonopore respectively. The endophallus opens into the edegus through an aperture. The phallotree. The phallotheca is a tubular sheath laying around the edegus, and it is formed due to enfolding of a distal part of the phallobase. In the case of edegus is reduced partly or completely, then the phallic tube is called the theca and its inner lining, the endotheca. The edegus is a tubular structure in the majority of insects. So it is completely sclerotized except the terminal membranous part called the preputial sac or vesicle. The distal extremity of vesicle is armed with spines, small plates or slender processes, the titulitors. According to Matsuda in 1976, the penis of insects can be grouped into 4 types. Edegus type penis. It is found in Thysanura, Hemiptera, Coleoptera, tubula. It is formed from edegus. Then phallomere type penis. It is found in Hemimetabola, Blatoidea, Mandoidea, some Placoptera, Phasmida and Orthoptera. The penis consists of 2 to 9 phallomeres. Then endophallus type penis. It is found in Placoptera. Due to loss of phallomeres, it is formed from endophallus. Then phallotheca type penis. It is formed from the phallobase or phallotheca, and endotheca found in Sarcoptera, Thysanoptera and Malophaga. The homology of different types of penis cannot be made. The penis of insects is a complex structure which is composed of phallobase, endophallus, edegus, phallotheca and or endotheca. Then the periphallic organs. They are peripherally located around the intromittent medium phallic organ complex in Thysanura, gonopods rising from the 9th segment, and in Pterygota, various movable and immovable appendicular processes represent the periphallic organs. They are commonly called as the harpagons or claspers. In Pterygota, they are lateral in position and are modified into slender processes, broad lobes, small hooks or falciform arms. They serve as claspers. They are provided with muscles. They are reported in Ephemeroptera, Hemiptera, Neuroptera, Mecoptera, Lepidoptera and Diptera. The unmovable accessory periphallic organs widely vary among different groups of insects. Then Thysanuran periphallic lobes. The gonopods, they are the paired structures as appendages of the 9th abdominal segment and are closely associated with the penis. The gonopod consists of a large broad exopodite and a slender distal stylus. Then in case of female genitalia, the ovipositta, that is the egg-laying tubes. The female genitalia mainly consist of a simple egg-laying tube known as ovipositor. It is basally communicated with the female gonopore, which is located on either 8th or 9th segment. It is not gonopophyseal origin. The paired gonopophyses can develop on the sterna of the 8th and 9th segments from their tubular structures. It is found in most of the Thysanura and Pterygota, while it is absent in Columbola, Diplura, among Pterygota and Ephemeroptera, Odonata and other Lepidoptera like that. Then substitutional ovipositor. The tubuliferous Thysanoptera, Mecoptera, Lepidoptera, etc. bear substitutional ovipositor in the absence of true ovipositor. The substitutional ovipositor represents simply a telescopic tube. It is formed due to modification of the posterior abdominal segments into a tubular extension which acts as an egg-laying tube like a true ovipositor. In some insects, particularly fruit flies, the last abdominal segment modify into tapering tubular structures which is pointed at the end so that it may pierce the skin or rinse off the fruit for ovipositing the eggs. And structure of a typical true ovipositor. The gonopophyses or valvulae in Pterygota. The ovipositor is formed as a complex of paired appendages originated from the 8th and 9th segments. The appendages are known as either gonopophyses or valvulate. They occur in 3 pairs. The paired appendages of the 8th segment are called the anterior or ventral or first valvulae. They develop as the median outgrowths of the coxal area. Similarly, Those of ninth segment are called the dorsal or posterior or second valvulae. They are modified from the mesial or endite process. The coxopodite of the ninth segment form the lateral or third valvulae, which ensheath and anterior and posterior pairs of gonapophyses. They grow as the distal outgrowths of the coxopodites. The well-developed ovipositor can be seen in Orthoptera, Hemiptera, Thysanoptera, and Hymenoptera. The lateral valvulae may bear styli in some insects like Hemiptera, Coleoptera, and Neuroptera, while in others the styli are absent, like Blatteuria. The coxite of segment 8 is called the first valvifer, bearing the anterior valvula. Similarly, the basal or coxal area of ninth segment is called the second valvula, bearing the posterior valvula. The paired second valvulas are often united ventrally by a narrow bar. The ovipositor is composed of a shaft and a basal apparatus. The shaft is made up of first and second pairs of closely arrested elongated processes, the valvulae. The basal apparatus consists of two pairs of lobes or plates, the first and second valvifer, which provide support to the shaft by the bases of the valvula. The proximal parts of the valvulae may differentiate as the ramming that fuse with respect to valvifer. The lateral valvifer form a third pair of blades in the shaft in Orthoptera. The muscles of ovipositor are inserted into valvifer. Styli never occur on coxopodites of the 8th segment, and those of the 9th segment are lost during the last adult molt, except in some Odonata. So, Hymenopteran ovipositor is specific in bearing articulation of the second valvifer with the first valvifer, if not with the 9th terga. The shaft is pointed and slender, elongated, and provided with lateral ridges. It is formed from the first and second valvulae and ensheathed with the third valvulae. The two second valvulae are united to each other by a median membrane. Then, in the stinging hymenopterans like bees and wasps, it loses its egg-laying function and is inverted into a poison-injecting instrument without much structural changes. Second valvulae modify into a median styles, and first valvulae into the lancets, which are major mobile elements of the sting. The valvulae on the bases of lancets drive the poison liquid through the channel of the united second valvulae, from which it escapes ventrally between the tips of the lancets. So we mentioned some important aspects related to the external morphology of insects. We learned about head segmentation, structures of head, sutures, and along with that sclerites of head. Then antennae, outparts, thorax, wings, and legs. And in this particular session, we learned about the abdomen in detail, along with some external genitalia of male and female in case of an insect. Along with that, pregenital and postgenital segments we mentioned, skeletal composition of this abdominal segments we mentioned. So with this, I'm gonna stop about the insect morphology. Later we will be learning about the insect anatomy in the sense digestive system and all other sort of important systems and their function. So thank you for listening this video.